Good afternoon. This is Dr. and I am very happy to inform and to let everyone know on the panel member that Mr. Himanshu, who is going to be presenting his open defense for his PhD, and myself, Dr. Vishnu Kirti, Department of Microbiology, Assistant Professor, and I am the chairperson of this particular PhD open defense. Along with me is Dr. Alok Mishra, who is handling the Department of Microbiology as Assistant Professor, and next to him is Dr. Anuprita Ray, who is the supervisor of the candidate, Mr. Himanshu, and at the last, chief guest of the session, Dr. Gurinder Walia, sorry to say. And now I would like to give a brief introduction regarding our external panel member. Respected Dr. Gurinder Kaur Walia, on behalf of our institution, I extend my warmest welcome to Dr. Gurinder Walia, Professor of Zoology and Environmental Sciences at Punjab University, Patiala. Dr. Walia is an eminent scholar and researcher with over 3 decades of academic and research excellence. She brings with her an impressive portfolio of her accomplishments. More than 105 peer-reviewed research publications, authorship of book chapters with prestigious publications through including CRC Press, and substantial contributions to molecular cytogenetics, toxicology, and molecular phylogeny. Her expertise spans diverse areas such as odontology, cytogenetics, and DNA barcoding, fields where she has earned international recognition. She is the fellow of South Asian Council of Odontology and has been honored as an outstanding odontologist of India, a testament to her pioneering contributions to the field. Mr. Himanshu, you are quite privileged to have such a distinguished expert evaluating your doctoral research. Dr. Walia, we look forward to your insightful evaluations and your valuable feedback towards Mr. Himanshu. Thank you, Dr. Walia, for your gracing this Viva Voice, and we wish to give a productive evaluation session through your part.
Thank you very much.
Over to you, Dr. Walia.
Thank you very much for the brief introduction about me, but there is no need, I think.
That's okay, ma'am.
But Himanshu, you have done a good work. I am here to listen to your presentation for the Viva Voce exam. You can start.
Okay. Thank you, ma'am. So, very good afternoon to all of you present here. So, I am Himanshu Jangid, PhD Scholar, Department of Microbiology. Today here in front of you to present my research work. So, the title of my research work is Biogenic Synthesis of Silver Nanoparticle Used in Soil Fungi and Against Carbapenem-Resistant Escherichia and Klebsiella pneumonia. And I am doing my work under the supervision of Dr. Anuprita Ray, Assistant Professor, Lovely Professional University, Jalandhar, Punjab, and under the co-supervision of Dr. Gaurav Kumar, Professor, Amity University, Jaipur, Rajasthan. So this is the table of content of my presentation, which start with the introduction of my proposed research work. So as we can see that these infections, whichever caused from bacteria, fungi, parasitic, any kind of infection has been a problem, and infections which has been caused from multi-drug organism which are resistant to a certain more than one group of class of antibiotics has been a major concern. And then as we see from the data, that WHO has also recognized antimicrobial resistance as a silent pandemic that threatens the success of the modern medicine as of now. And as we see that WHO has also published a global priority pathogen list in which he has listed bacteria, around 12 bacterial species, out of which, like if you name Pseudomonas, Klebsiella pneumonia, Acinetobacter baumannii, these are one of the major problematic microorganism as we talk about. And if we talk about the data as WHO has published, so he has published certain data in which he has shown the loudest. So may I start?
You can continue.
Ma'am, continue. Okay, thank you. So So as we see the WHO data, some statistics has been published by WHO on bacterial infection, resistant bacterial infection, which has when we seen that about 1.27 million deaths and contributed to around 4.95 million deaths worldwide in 2019, as report published by WHO in 2021. And more recent surveillance data has also shown that about 4.71 million deaths globally occur, and with 1.4 million that directly attributed to the drug resistant bacterial infection. And as we move forward, as we have taught about the multi-touch organism, in this class there is class of antibiotic which is known as carbapenem, and it has been used as a last line of resort. It is an antibiotic which we given to the patient in case of heavy multi-touch resistant bacterial infection. And from last 2 decades, and morely from the last decade, as we seen that in this group of antibiotic the resistance has also been pronounced. And as we seen the data that CRE infections have shown a mortality rate as high as 35% in clinical setting and across the low and middle income countries. And because of these resistant infections, the main problem is also like it will affect the countries, multiple countries economically, as the cost of medicine increases, the cost of hospitalization increases, which also affect the country economically. And as we move forward, we can see that there are certain, if we are talking about the bacterial infection now, then there are certain antibiotics which are used to treat carbapenem-resistant infection, but those antibiotics and even combination therapy which are using right now are also showing failure. And multiple novel drugs we can talk about, modal approach, therapeutic approach we can talk about, has also not becoming that much popular. So in our cases, we use the help of nanotechnology, in which we have synthesized silver nanoparticle. And basically, why silver nanoparticle? As we know that silver nanoparticle has been known for their broad spectrum antimicrobial activity against many gram-negative bacteria, many gram-negative multi-touch resistance bacteria. And as of now, I taught about the recent publications are also come in which they have shown activity of this silver nanoparticle, biogenic silver nanoparticle against carbapenem-resistant bacteria even. Initially, it is not happened when we started the work. And as we move forward, I have taught about the word biogenic. So in our cases, we have synthesized silver nanoparticle from the biological source, and the biological source which we have used is the soil fungi. This is the agricultural soil we have collected from different sites in the near the Fadwara region, and from that we have isolated soil fungi, and from that we have synthesized silver nanoparticle and checked their antimicrobial activity against our pathogen. So as a conclude, we can see that this fungal derived AGMP emerges as a sustainable, potent and biological driven nanotechnology solution to address the global crisis of this MDR infection. So as we move forward to the literature review part, so in the literature review of biogenic synthesis silver nanoparticle, we can see that many sources has been used for synthesizing silver nanoparticle. As I talked about fungi, plants, bacterial, each has their advantages and disadvantages. And the next is biogenic synthesis silver nanoparticle. As we can see that from the biological sources, many nanoparticle has been synthesized, silver, zinc, copper, and in our cases we have used silver nanoparticle, which is the reason it is shown the broad spectrum antibiotic against gram-negative pathogens. The next literature is about biosynthesis of silver nanoparticle mediated via fungi, in which we have shown that many fungi like Aspergillus, Candida, Penicillium has been earlier reported to synthesizing silver nanoparticle. The extracellular components in this fungal extract has been act as a capping agent, reducing agent, which help in the formation of the silver nanoparticle, as we seen in the literature. Then this is also shown that synthesis of silver nanoparticle via fungi. And the next literature review is about the mechanism of these biosynthesis and factor affecting biosynthesis. So as of now, there are not much literature has been published regarding the how the silver nanoparticle is synthesized, the mechanism behind it. And in few papers, we are able to find that there are certain enzymes like NADP, NADPH, NADP dependent reducer have been involved in somehow in the reduction of the silver nitrate, which form the elemental silver, and further which forms with the capping and biomolecule capping from the fungal ester from the silver nanoparticles. And next is the carbapenem-resistant enterobacteriaceae, as this is my main work on the bacteria. And in this we have literature, we have find that there are certain enzymes which are involved in the carbapenem-resistant. There are basically two types of mechanism which I have seen in the literature review. The one is the enzymatic mechanism behind the carbapenem-resistant, and another one is the non-enzymatic. In the enzymatic, we can see that there are certain carbapenem enzyme which hydrolyzes these antibiotic, which are NDM, KPC, OSA, IMP. These are the carbapenem enzyme which are regulated by genes like BLA-NDM and BLA-KPA, which are involved in this Carbapenem resistance caused by enzymatic, and in non-enzymatic we can see certain mechanism like porine mutations, membrane permeability. These are the mechanisms which are involved in the non-enzymatic carbapenem resistance. And this is a literature which are found regarding the certain enzymes, certain mechanisms which are involved in this. And the last is the literature in which we have seen that certain silver nanoparticle we have found that showing antimicrobial activity against gram-negative pathogens like E. coli, Escherichia, Pseudomonas aeruginosa. And the recent study from last 25 to 220 paper, I have also found that there are certain which have reported against carbapenem resistance gram-negative bacteria also. So this ends my review literature, and this is the research gap of my proposed study. So the rapid rise of antimicrobial resistance in Enterobacteriaceae, particularly Escherichia coli and Klebsiella pneumonia, has become a major global health concern. These pathogens are responsible for several infections such as bloodstream infections, wound infection, urinary tract infection and pneumonia, and have acquired multiple resistant mechanisms that undermine the efficacy of most available antibiotics. The situation is specifically critical in case of Carbapenem-resistant Enterobacteriaceae, as carbapenem are considered as last line of defense for treating multi-drug resistance gram-negative infection, as I discussed earlier, due to their high mortality rate, as we seen in the literature part and the introduction I have write up, and treatment failure and the rapid spread. The WHO has classified CRE under the critical priority group pathogens requiring immediate attention and urgent development for the new therapeutic strategy. Despite the severity of this threat, there is a limited research, specifically Biogenic synthesized silver nanoparticle, which serve as an alternative strategy for carbapenem-resistant Escherichia coli and Klebsiella pneumonia. Although silver nanoparticles are widely recognized for their broad spectrum antimicrobial properties, few studies on fungal mediators, especially Biogenic synthesis for targeting CRI, have not reported yet now. The fungal represented as a promising source for reducing and stabilizing biomolecules, and published study increasing highlight their potential to generate eco-friendly, stable and highly active nanoparticle. However, their application against the particular carbapenem-resistant Enterobacteriaceae remain underexplored, creating a clear gap for the study to aim to address. So this is the purport objective of my proposed research work. So the first objective is my biosynthesis of silver nanoparticle using soil fungi and their preliminary characterization. My second objective of my research is the assessment of antibacterial activity of biosynthesized silver nanoparticle against carbapenem-resistant Escherichia coli and Klebsiella pneumonia. My third objective is the exploration of mechanism by which the antimicrobial activity of this synthesized silver nanoparticle and the toxicity evaluation of those biosynthesized silver nanoparticle. And my last objective is the polyphasic characterization of potential fungi and physiochemical characterization of the potential silver nanoparticle, the most potent nanoparticle which we get from our study. So this is my objective one, in which I have isolated fungi. I basically initially first I collected different soil sample region near the region forward agriculture soil, and then I isolated different fungal sample from them, and then I've used those fungal sample for isolating silver nanoparticles. So this is a soil sample collection, and this table we can see there are different geographical detail and classification of soil sampling site I have done, which I have stored the sample like S1, S2 and S3, and these are the location, latitude and longitude of each the location. And the right side we have to see that these are representative images of the two sites which I have taken. And this one is the isolation of fungi, as we have done the isolation on PDA plate supplemented with bromophenol to avoid the bacterial contamination, and incubated them at 28 degrees Celsius. So these are my sample number. From each sample I have gotten certain amounts of isolated. The number of isolated from each sample has been mentioned here, and this is the isolated coding which I have given for each sample, like S for sample and I for isolated, and these are coding and number from each sample isolated which I have gotten. And these are the representative images from my each sample. These are the number of coding and these are the images. The right panel here shows the reverse. The right panel here shows the reverse side of the river view of the PDA plate, same PDA plate, and the left side is the front view of each isolated. So from sample 1 I have isolated 6 isolated. These are the representative images from each. The figure 2 is the representative images from my sample 2, the 3 isolated which I have gotten, and from sample 3 I also getted 3 samples. From sample 4 I have gotten 5 total isolated, and representative images for each has been shown here. And these are the 4 isolated which I have get from my sample 5, and these are the last 4 isolated which I have gotten from my sample 6. This make a total of 25 morphologically distinct isolated which I have gotten. Now next part is the synthesis of silver nanoparticles. So initially what we have do, we have prepare intracellular and extracellular extract from each fungi. For preparing that, we have incubated them at PDB for 28 degree for 5 days, and the biomass was separated to obtain the extracellular extract. And for the intracellular extracted, what we have done, we have harvested the fungal mat which were washed again with for intracellular extracted, we have harvested the fungal mat, was washed and suspended them in sterile water and incubated them under shaking condition to induce osmotic release of intracellular metabolite, and the filter was collected and stored at 4 degrees Celsius. And now the biosynthesis part, in which we have used both intracellular and extracellular fungal extracts. So for the synthesis process, what we have done, we have taken the reaction mixture have included 1 millimolar AgNO3 in the ratio of 19:1, and we incubated them at 28 degrees Celsius plus minus 2 degree on a chater for around 48 hours. And after 48 hours, we have checked for is there any color changes which have observed, and based on the color changes, preliminary characterization which we have done based on the two things. One is the color changes, another part is the UV spectrum, which we have taken from the range which we have taken in the range of 200 to 300 nanometer. These are the representative images of our reaction mixture synthesized silver nanoparticle. And here the images all shown here for the sample 1, 2, in which on the left side we can say these are the images from the intracellular extract, and the sample 1, and the right side is the extracellular extract. And we see that A is our 1 millimolar AgNO3 solution, the B is our fungal extract, intracellular in case of intracellular synthesis and extracellular in case of extracellular synthesis, and C is our reaction mixture after 49 hours. And in certain isolated like in S1I4 intracellular, if there any color change induced in any of the sample in C, we can say that the color changes from light brown to dark brown. So these are the representative images from the each sample and each extract. These are the representative image from sample 4, 5 and 6. Then next, after we done the preliminary characterization, the color changes, we have done the UV spectrum for each from 200 to 800 nanometer, and we can see that there is a specific peak which we have observed between the range of 390 to 450 nanometer, which further confirm as the successful AgNP formations. So these are the UV graph for our sample 1, including both intracellular and extracellular. And this is for our sample 2. In these cases, we will see the C, that's the S2I1 and S2I2 intracellular. There is a peak which we are observed within the range. The peak we have observed for each isolated has been different. Like in certain, we have observed the peak around 390, 412, 418. This specific difference has been observed due to the size and the aggregation of the silver nanoparticle which I have observed. So these are from the sample 2 UV spectra, and these are UV spectra for our sample 3 isolate. These are UV spectra for our sample 4 isolates, which is shown the range between 200 to 800 nanometer. And these are the UV spectra for our sample S5 isolate, all including intra and extracellular. And these are from our last sample 6. And this is the table in which we can say we have named all the isolated, the extract which we have, and the color changes. If we see in the case of S1I4, the intracellular and extracellular extract show a dark brown color, and the peak which we have observed in the range of 418 to 426 nanometer. And as says, the shift which we have seen from the range we have seen from 450 to 390 nanometer is due to the size and the aggregation of the nanoparticle. If they are closer to the 390 nanometer, then we can say the size are small and aggregation is less. And if they are above like 430 and 440, then we can say the size of the nanoparticle are larger and aggregation. So in this images we have seen the S1I4 and S3I2 have shown the silver nanoparticle synthesis, and H2I2. And this is the table images for the remaining, in which we seen that certain peak for S4I3 showing 391 nanometer, and S5I1 showing the synthesis for the both the intracellular and extracellular, and these are the peak which you have observed in 436 and 450 nanometer. So here is and our objective 1, in which we have collected different soil samples, basically 6 soil samples. From that we have isolated 25 morphological distinct fungi, and from that we have made intracellular and extracellular acetate, and from those we have synthesized silver nanoparticle. So total of 8 silver nanoparticle we have synthesized, out of which 6 we have used, because 2 are from both intracellular and intercellular acetate. And our next objective, now we have now synthesized the silver nanoparticle. Now our next objective is the assessment of the antibacterial activity of this biosynthesized silver nanoparticle against Carbapenem-Resistant Escherichia and Klebsiella pneumonia. So here first what we have done, we need the bacterial isolated of E. coli and Klebsiella pneumonia which are Carbapenem resistant. So for that we have contacted the PIMS, Punjab Institute of Medical Science. From there we have collected the sample with the pure IBSC approval from the institutional biosafety community, Lovely Professional University, which is done by the DBT India. Then next, after we have collected a sample and we have transported them here, now what next we have done, we have recharacterized those bacterial isolated, as those bacteria has been characterized as from PIMS, but we have to recharacterize them to sure these are those bacteria, there is no certain contamination happen. So for the recharacterization, we have recharacterized those bacteria, E. coli and Klebsiella pneumonia on EMB. We have done gram staining and we have done the standard biochemical test for them to confirm their identity. So these are the results from our biochemical testing and gram staining and street on the EMB. On the left side, the green metallic sheen has been shown the characteristics of E. coli, and then mutoid colony has been seen, pinkish mutoid colony, dark pinkish to purplish. And so on the right is of Klebsiella pneumonia, and this is the table which show the biochemical characterization of clinical isolated. For E. coli and negative, all the E. coli and negative are both are negative. The IMBIC we have done, TSI and urease oxidase catalase and done. We have confirmed them with the pure literature that these are consistent with the E. coli and Klebsiella pneumonia. This biochemical characterization. And our next part, now we have checked there that these bacteria which have collected from PIMS are E. coli and Klebsiella. Now we have to confirm one more thing before doing our antibacterial testing, that is the confirmation of their Carbapenem resistance. So for doing that, we have purchased the disc of imipenem, meropenem, 10 microgram per ml disc we have purchased, and we have for the checking the Carbapenem resistance, we have checked them using the disc diffusion method, which are as per the guidelines of CLSI, and we have made the standardized 0.5 MC violent suspension which are incubated in the Muller-Hinton agar, and imipenem and meropenem discs were applied, and after 37 we have calculated and measured the zone, and we have measured them with the CLSI breakpoint for these antibiotics. So this is my result. As I taught about the CLSI guideline, as per the CLSI guideline, the breakpoint zone of inhibition for meropenem and imipenem is the 19 mm zone. Below 19 mm zone they consider as resistance, and in our case of E. coli and Klebsiella pneumonia, we can say the zone is below the 19 mm in case of both E. coli and Klebsiella pneumonia. In this we can see the A disc is for the imipenem and the B one which we use for the meropenem. And if you see in the case of Klebsiella pneumonia, the imipenem disc has been not showing any zone. So we can also confirm that the isolated which we are collected, they are not resistant, they are highly resistant to these antibiotics, which we can see here. And as we confirm the Carbapenem resistance, we have recharacterized the bacteria. Now we are ready to perform the antibacterial activity using our biosynthesized silver nanoparticle. So for doing the antimicrobial activity, we have used the agar well diffusion method. So for that we have again prepared the 0.5 ml standard suspension which was swabbed on the MHA plate and which are loaded with the AGMP. Here for the initial preliminary screening of our nanoparticles, we have used the concentration 1 mg per ml from our 6 fungal isolated, and the inhibition zone were measured after incubation. And as we can see here in our agar well diffusion method, we have used our 6 nanoparticle which have biosynthesized from the fungi, 25 fungal isolated. These are labeled here S1I4, S2I2, S3I1, D is the S3I3, S4I3 and S5I1. These are the 6 isolated. The standard concentration we have used here is the 1 mg per ml for each. And the left side the image is for E. coli and right side is for Klebsiella pneumonia, and here we can see the zone of inhibition which we have measured. So for the case of S1I4, it has shown the highest zone which is around 24.33 mm plus minus 1.52. And for the Klebsiella pneumoniae, it is also shown the nearly same zone at 25.33 plus minus 2.08, and the remaining isolated have shown, but they are comparatively lower to compared to S1I4. Based on this interference, we have considered that our S1I4 isolated has been the most potent isolated, and the star has been shown that we have applied the ANOVA. In this ANOVA, we have checked that S1I4 is comparatively greater, significantly greater than this remaining 5 silver Biogenic silver nanoparticle which we have used. So here the most potent silver nanoparticle which we have getting there is the S1I4. We have tested them at different concentration. Last concentration which we have 1 mg per ml. So we have reduced the concentration to that at lower concentration. It is also shown the potent antimicrobial activity. So for that we have used the concentration 0.5 mg per ml, 0.25 mg per ml, and 0.125 mg per ml. At the C, we have used the positive control meropenem. It is in the concentration of 0.1 mg per ml, and the E is our negative control. That is our sterile distilled water, because that is the water which we have used in the preparation of our silver nanoparticles and the antibiotic. So in this cases, the zone we have seen, in case of E. coli, when we reducing the concentration, the zone is also reducing. But in case of Klebsiella, we can see that when we reducing the concentration, the zone nearly minimized. The zone is no clear zone has been shown in case of 1.125 and 0.51 mg, which says that at a certain condition that is also not showing certain zones. And in this, what we have collected, the samples we have collected from PIMS. So for both E. coli and Klebsiella pneumonia, we have collected 10 isolated each. We have collected that because we have want to know that our nanoparticle has been showing the same result in different, because in different isolated the resistance will be different. Some are highly resistant, some are differently resistant. But our silver nanoparticle has been showing, if you can see in this picture in the E. coli, at different concentration our silver nanoparticle has been shown a consistent zone which is around 22.32 to 20 range, and in case of positive control, the zone is also consistent, and it is 13 mm zone and 14 mm zone which I seen, and as per the CLSI breakpoint below 19 mm is resistant. So we can say in our case these bacteria are highly resistant. And this is for the Klebsiella pneumonia. It is also show the significantly larger zone in these, all consistent along all the isolated. And then after doing the antimicrobial well diffusion method, we have done the MIC and MBC from our potent nanoparticle, that is the S1 and I4. So for doing the MIC, we have used the microbroth dilution method, which is also followed by the CHLA guideline. We prepared AGMP stock solution between the range of 5 to 200 microgram per ml. These are the final concentration which we have used, and meropenem control was also prepared in the same concentration, and we have prepared them in the 96 well titer plate and incubated them with the standardized bacterial suspension, and which is also prepared as per the CLSI and UTAS guideline recommendation which have used throughout the method. And the plate were then incubated at 37 degrees Celsius, and the lowest concentration showing no visible turbidity was recorded as our MIC. And for the MBC, we have determined by subculturing 10 microliter from each MIC well, showing no visible growth on 2 MHA plate, and this plate also incubated at 37 degrees Celsius, and the lowest HGMP concentration producing no tolerant growth was recorded as the MIC, and all these assays have been proposed has been done in triplicates. So this is the table which have shown the MIC and MBC of my biosynthesized silver nanoparticle, and I have compared them with the standard antibiotic meropenem which we have taken. So in this case, if I talk about the CLSI guideline first, so as per the CLSI guideline, the breakpoint MIC for meropenem is 4 microgram per ml. So above 4 microgram they are resistant, and in our cases you see that in case of E. coli, the MIC for meropenem is 25 and the breakpoint is 4. So we can say that here also is correctly shown that our isolate are highly resistant, and in case of Klebsiella pneumonia it is higher as a 60 microgram per ml. And if I compare my silver nanoparticle with the meropenem, so my E. coli has shown the MIC at 5 microgram per ml, which is around 5 times less than 25 microgram compared to meropenem. As I compared with the Klebsiella, it is also reducing 2 times reducing, which have shown that our nanoparticle at very lower dose has been shown the good antimicrobial activity compared to these antibiotics which has been used for treating this Carbapenem resistant infection. And as we move further, now we have in our objective 1 we have isolated fungi make silver nanoparticle. And our objective 2, we have tested all our silver nanoparticle and find our most potent silver nanoparticle. Now next what we have done, that we have studied the mechanism by which it induces the antimicrobial activity in this organism, in this microorganism, and also we have done the toxicity evaluation of it, as it is very important. As you know, as I say that my MIC is 5 microgram per ml, but my toxicity is also higher, then it doesn't make any sense because I am not able to make any therapeutic dose with this nanoparticle further in our future studies. So that's why we have done that also toxicity evaluation. So in the mechanism we have done the 4 mechanism studies. First is our growth curve analysis, in which we have studied bacterial growth kinetics, which were measured in Muller-Hinton broth with 4 flasks we have taken. One is our blend, the second one is our bacterial control, the third one is our meropenem at half of MIC concentration, and fourth one is our silver nanoparticle, the most potent nanoparticle at half MIC concentration. The half MIC concentration we have taken here because we have not done the growth here. In all other experiments we have used the MIC concentration because the test has been applied after the growth has been done. If I use MIC concentration here, the growth has been not occurred and I am not able to study the bacterial kinetics. So that's why I use the half MIC concentration here. And we have measured the OD at 600 nanometer were recorded every 2 hours for 30 hours, and mean OD value triplet were plotted versus time to compare lag, exponential and stationary phase. So in this result we can see that the left one is the E. coli and the right one is the Klebsiella pneumoniae. If I talk about the E. coli, that blue line is for the AGMP, the green one is for meropenem, and the red one is for the control. The control has been shown the standard growth kinetics which was seen for the E. coli, but in case of AGMP in my nanoparticle, it had extended the lag phase up to 14 hours. Up to 14 hours it has extended the lag phase, and after that the growth has been started, which has shown the inhibitory effect of my silver nanoparticle. And in case of Klebsiella pneumoniae, we are also seen that the lag phase has been extended up to 14-15 hours. And if I compare it with the antibiotic, the meropenem is only able to extend the phase up to 6 hours. In case of my nanoparticle, they were extended up to 14 hours, which also overall reduce the bacterial growth. And the next assay which we have performed is the nucleotide leakage assay, in which we have tested that there is a cell membrane damage. If there is cell membrane damage, there is a leakage, release of nucleotide in the medium, which we have measured at 260 nanometer. So for this also we have prepared standard bacterial suspension, which were treated with AGMP at each isolated at MIC concentration as I talked about, and meropenem and water control we have also taken to compare them, and they are incubated at 37 degree plus minus 1 degree, and the sample were hourly taken at each interval of 1 hour, and each interval we have centrifuged them up to 8 hours, and a supernatant has been which we have used for take the absorbance at 260 nanometer. And these are the results which you have seen as same. The left one is the E. coli and the right one is the Klebsiella pneumoniae. The red one has the control. In both cases you can say that it doesn't show much of the damage. The minimum OD which will increase is due to the shear stress, as each hour we are doing the centrifusion. Because of them there are certain shear force which are applied to the membrane, because of them there is certain increase, of the OD. But if we compare them with our antibiotic, the meropenem which we have used, and our silver nanoparticle, then the damage is very pronounced, and there is the leakage, the nucleotide leakage has been observed and recorded at 260 nanometer. And here we also say that as compared to antibiotic, our silver nanoparticle are far superior in doing the cell membrane damage. And the next one is the nucleic acid damage assay, in which we have seen that our silver nanoparticle are also able to damage genomic DNA or not. So for that we have prepared our AGMP treated sample and control cultures, and they are treated at MIC value, incubated for 20 hours, and then we extracted the DNA using the C-type phenol chloroform protocol, which included RNase and ethanol precipitation too, and the resuspended and TE buffer, and analyzed at 1% agarose gel at 100 volt to detect the DNA degradation via gel electrophoresis. So this is the image which agarose gel electrophoresis image which showing genome DNA integrity of E. coli and Klebsiella pneumoniae. Here we can say the lane 1 and lane 2 are for the E. coli. The lane 1 is the E. coli control and lane 2 is the E. coli treated. The lane 3 is our DNA ladder, and lane 4 is the same Klebsiella controlled, and the lane 5 is the treated. So both cases in lane 2 and lane 5, we can say there is not much DNA fragmentation occur, which makes us to infer that our nanoparticle not inducing much of the genotoxicity to the, not doing the much damage to the DNA. And if you see, there are multiple bands which are shown in the lane 5 in case of Klebsiella pneumonia treated, and it is happening because in few literature we have found that Klebsiella pneumonia, when we do the genomic DNA isolation for Klebsiella pneumonia, due to large plasmid and large DNA, there are certain plasmid terms alongside the DNA isolation, which have shown and supported by references, which has also shown that in genomic DNA isolation, sometimes plasmid has been also isolated, also found, and which is shown in the gel for this image. And the next one is the last one. We have checked the cell membrane damage, we have checked the growth kinetics, we have also checked the DNA damage. The last one we have checked the cell wall, is the damage in the cell wall. As we have taught about the carbapenem, so carbapenem mechanism is basically cell wall damage. And in case of our nanoparticle, it also inducing cell damage, which will shown in the further result. For the experimental setup, we have take the treated and controlled bacteria which are exposed to EGMPT at MIC concentration for 3 hours. Then we wash them, fish in 2.5 glutaraldehyde, dehydrated them through the ethanol using the sequential ethanol process from 10%, 30, then 50, then 70, 90, and 100 we are 2 times, and then last drying we are doing with the HADMS, 2 concentration 50% and 100%, and then we use the air dry our sample on a slide, is coated with AU and imaged by FSM to compare the surface morphology and the membrane damage. So here in the images we can see that our A, B, C are images for the E. coli, and our D, E, F, the lower panel images are for the Klebsiella. The A and D images, you are seeing the intact bacterial morphology, because these are the control sample, and after we treated them with our EGMP, you are seeing there are some very much damages which are shown that completely rupture the cell and their cell wall and is in damage, which is seen in the followed by image of B and C for E. coli and the E and F for Klebsiella pneumonia. And the B and D images are at a lower scale, and we have actually thought of doing it at a higher magnification scale today how much damage is and see. And the C images we can see for the E. coli, they completely rupture the cells, and the content of the cytoplasm has also come out of the cell, which is seen in this same image. And this is the summary of the mechanism of antibacterial action of our biosynthesized EGMP, which in with few observation we can say in case of growth kinetic suppression, we can say that at some MIC AGMP cause a pronounced delay in growth initiation and reduce overall burst compared to antibiotic, the meropenem which is used, and the control group. In case of membrane leakage, membrane disruption, we can say that treated cell exhibited significant nucleotide release, whereas antibiotic treated cells show moderate leakage and controlled show none. In case of genomic DNA damage, we can say that electrophoresis revealed smeared fragmented DNA in E. coli, which is very slight, and while Klebsiella pneumonia displayed distant banding linked to plasmid co-isolation rather than the typical DNA fragmentation. And in case of cell wall disintegration, which we have done by FSM, we can observe that FSM images revealed irregular shape, ruptured surface, collapsed cell wall in treated cell, and untreated bacterial cell retain their smooth intact morphology as we seen in the previous images. And the next one is the MTTT cytotoxicity assay. So we have checked the cytotoxicity of our biosynthesized silver nanoparticle using the cell line 3T3L1 mouse fibroblast, which is the MTTT assay, followed by the standard protocols. So initially we have cultured the cell wall culture in the EM medium, which are supplemented with 10% FBS and seeded to around 1 × 10⁵ cell per ml in the 96 well titer plate for around 24 hours. Then we prepare our straw solution in the AGMP in the concentration of 25 to 400 microgram per ml, which are prepared in the serum free EMBM and were added with the EMBM only cells as controlled. And after 72 hours of incubation and 5% CO2 cells were washed, treated with the MTTT reagent, which we have prepared in the concentration of 0.5 mg per ml for 4 hours, and the formazolin dissolved in DMSO, and the absorbences were recorded for each at 570 nanometer. The cell viability was calculated relative to the untreated control, and the dose response curve was generated. So here the images or the phase contrast images, microscopic images of dose dependent cytotoxicity of silver nanoparticle. This A, B, C and D have all the different concentration which I have mentioned down below, from 25 to 400 microgram per ml, and as we go higher the concentration, we see the progressive reduction in the cell density along with the morphological alterations such as rounding the membranes, blabbing and detachment, which are observed in increasing concentration, which has indicated as a dose dependence cytotoxic effect from our silver nanoparticle, biogenic silver nanoparticle. And this is the toxicity, this is the data we have plotted for the different concentration, and as we increase the concentration, the cell viability has been decreases, and as per this graph we can say the IC50 which we have calculated is around 100.06 microgram per ml, which is comparatively 4 times, which is 10 times lower than compared to our MIC, which is 5 microgram per ml for E. coli and 25 microgram per ml for the Klebsiella pneumonia. And this is our final objective, last objective, in which the potent nanoparticle which we have gained initially from the potent fungi, we have characterized both of them. So initially first we have characterized our silver nanoparticle, the potent silver nanoparticle, that is the S1I4 isolate. So for that we have done certain characterization like dynamic light scattering to get the hydrodynamic diameter, the size of a silver nanoparticle, zeta potential to share their stability. FTR analysis we have done to know the certain functional group has been involved in the biocapping synthesis, which are come from the fungal ester which have used for the synthesis of biosilver nanoparticle. The SRD we have done to know the purity and the crystallinity silver phase of a silver nanoparticle. TAM imaging we have done to know the surface morphology, size. SAM is also we have done to know the surface of our nanoparticle, and EDS elemental analysis we have done to know the how much silver is just biosynthesized silver nanoparticle, what is the elemental composition of a silver nanoparticle. So this is the DLS and zeta which have done. In this DLS which we can see that our silver nanoparticle showing a size of 24.55 nanometer, which is come under the as per the literature we have known that nanoparticle ranging from 10 to 100 nanometer has been known to show the potent antimicrobial activity, which are also shown here. In our case is shown the 24.55 nanometer size, and with the zeta potential we are able to know that our silver nanoparticle is moderately stable. As you say, the zeta potential come here is minus 23.66 MB. And this is the FTR spectra in which the green line has shown our fungal estate, and that silver nanoparticle is shown in the red of our isolate S1I4. The spectra here highlight the involvement of biomolecule from fungal estate which are involved in either reduction, capping, stabilization of our overall Biogenic silver nanoparticle. And this is the SRD pattern of silver nanoparticle. We have compared them with our standard JCPDS, silver JCPDS number, which is our number is 0.0783, and we have also certain peak which are shown in case of silver nanoparticle at a phase of 38 degree at 38.1, 44.3, 64.72, which we have from the literature we have found shown in case of silver nanoparticle. And this is the time image of my Biogenic silver nanoparticle. A and B images are showed at different magnification scale, and both images we are able to show that the silver nanoparticle I synthesized is less aggregated and showing the rounded and oval shape, spherical to oval shape silver nanoparticles, and the range which we have gotten from these images is around 29 to 34 nanometer. And this is our SAM images from the scale which have taken 100 nanometer. We are able to identify our nanoparticle has been ranging between the range of 20 and 40 nanometer, which are consistently showing in our results. And this is the EDS which has shown the elemental composition of the each element which are found in our silver nanoparticle. The silver peak has also been shown in this elemental composition. And the last one is the morphological and the molecular identification, which we have done using the ITS sequencing for the potential fungi. So for the morphological identification, the fungal isolated we have culture on the PDA at 28 plus minus 1 degree for 5 to 7 days, and we have observed the colony characteristics like what the texture, what the color, is there any certain metabolites produced, certain types of things, elevation, margin, surface texture, and the morphology we have done using LPCC stain mount, which are observed the hypal feature, conidiophore and spore morphology. For the ITS sequencing, we have isolated the genomic DNA using the CTAB method from PDA grown mycelium, and the purity was confirmed using the nanodrop and gel electrophoresis. The ITS reason, the primer ITS 1 and ITS 1 and 4 primer has been generated clean amplification that are purified and sequenced. The ITS sequence was analyzed using the BLAST and against the NCPI gene bank database, and the phylogenetic alignment and tree construction were further performed using the METHA 12 to confirm the isolated taxonomical identity. So this is the images showing the A and B is the images. The A is the front image and B is the back image, and here the texture which I am shown with the characteristics of the Aspergillus niger, and C is the LPCD mount image, which have shown the different biserrate fillets radiating from the globus vesicle shown upside along the round wall tonidia arranged in the teratostite head. The next table shown the colony morphological characteristics in which we have shown the colony morphology, the texture which is from the cottony to granular surface texture, the black texture which you have seen in the images. The growth pattern basically exhibited a central dense black zone which is surrounded by a white periphery, distinctly radiating fold but also visible. And for the conidiophore, they are hyaline, smooth walled, unbranched basically seen in the LPCB, arising from and foot cells and erect and stout. Vesicle, they are globulous to subglobulous, biserrate arrangement with fillet radiating across the entire vesicle surface. If I talk about the fillets, they are biserrated, covering the entire surface of the vesicle. Conidia, they are rough walled, globulous to subglobulous as the seen, brownish to black in mass and measuring about 3.5 to in diameter, which we measure from the certain software. And for the LPCB microscopy, it is a clear visualization of the clonidial head in which fillet and vesicle has been shown, and abundant black spore mass is also observed. This is the tree, phylogenetic tree, which we have prepared using the METHA software, in which we have submitted our isolated in NCPI, and this is the accession number PV203216.1, which is seen here, and the isolated which we have named is Aspergillus niger isolate AG3, which are closely branching and related with the other isolated, which we have seen in this image, with shown a strong bootstrap support with around 81% confirming its molecular identity. Bootstrap values basically range from hundreds of replicator and shown in this branch nodes. So as we done with all the 4 objectives, so this is basically a summary of my proposed research work. So the study began with the collection of 6 agriculture soil samples, which yield us 25 different morphologically distant fundal isolated. Then both intracellular and extracellular extract were prepared from these isolated, generating 50 independent silver nanoparticle biosynthesis reaction, 25 from intracellular and 25 from extracellular. These silver nanoparticle formation were initially confirmed through color changes and the spectral peak, which we observed in the range of 391 to 450 nanometer. Then among all the isolated, the extracellular extract of S1 iPhone display the strongest antibacterial activity, pursuing the largest zone of inhibition, which is around 24.33 and 23.33 for E. coli and Klebsiella respectively. And further, the best potent nanoparticle has been demonstrated antibacterial activity, even exhibiting low MIC, MBC value, which are significantly superior to the meropenem which we have used. And the mechanism studies are shown that AGMP exposure slow the bacterial proliferation by extending the lag phase and overall reducing the biomass. Mechanism assay also indicated membrane disruption through nucleotide leakage, genotoxic effect from DNA degradation, which is very less compared to the other. And FESM imaging revealed several morphological alteration, ruptured cells in the treated membrane collapse, surface irregularities which you observed. And by the toxicity testing using the MTTT assay, we confirmed that the antibacterial concentration required was much lower than the cytotoxic level, supported as a favorable safety profile. And the morphological analysis and the ITS sequencing identify as the potent fungal isolated, which one is our S1I4, which are identified as the Aspergillus niger with more than 99% sequence similarities. And the physiochemical characterization of our potent derived AGMP shows us that our nanoparticles were small, stable, bimolecule shape with an hydrodynamic size of around 24 nanometer and then zeta potential of around minus 23 MV, which shown as the moderate stability of a nanoparticle. FTR spectra revealed functional group which are associated in the which are found in the fungal extract and even present in our silver nanoparticle, which shows us that these fungal biomolecules are involved in reduction, stabilization of our silver nanoparticle and even the antimicrobial activity which we have seen. And SRD time, SAM, EDS analysis confirms the crystalline structure, spherical morphology and the elemental purity of our produced silver nanoparticle. This is conclusion which shown us that our soil fungi represent a robust, eco-friendly platform for AGMP synthesis, in which we have found 25 isolated and Aspergillus niger S1I4 comes as a potent and strongest nanoparticle producing capability. S1I4 derived nanoparticle are, as we say, small, crystalline. They are monodispersed. They show less aggregation. Biomolecule cap ensure stability and strong antibacterial property. AGMP show potent activity against our clinical isolated, which we have collected from PIMS, E. coli and Klebsiella pneumonia, which even surpass the Meropenem in camera, and just not surpass, just more superior compared to the Meropenem as we see in our studies. And the mechanistic assay confirm us that our silver nanoparticle has shown multi-targeted bactericidal effect, in which we have seen membrane damage, cell wall damage, even inhibiting bacterial growth kinetics overall and certain DNA damage. And the cytotoxic assay validated us that there is an acceptable biosafety dosage which we can make from a silver nanoparticle, which give us an establishing a promising therapeutic window. And overall in our study we can see that Aspergillus niger mediated AGMP offer as an sustainable high efficacy alternative to combat these CRE pathogens, which are Carbapenem-Resistant Enterobacteriaceae pathogens. And the future perspective I thought about that we have in our experiment we have not done the in vivo studies which we have done further, and further future perspective we have thought about we can use in different applications to prepare different ointment for certain wound infection. And in the future perspective I thought about we have not done the genomic studies of these Carbapenem resistant genes. If we thought about that, we can also do them in the future perspective studies. And the molecular identification which we done for Aspergillus niger further open us for door for more genomic and proteomic investigation to identify the exact fungal biomolecule responsible for nanoparticle synthesis, because in see that there are certain many extracellular proteins, enzymes, biomolecule present in the fungal extract, which is exactly responsible for the antimicrobial activity, which is responsible for the reduction stabilization. So noting that is a very key studies which we do in the future perspective of our studies. And this for the acknowledgement. So my deepest gratitude to my beloved parents, my mom and my dad, for their unconditional love, sacrifices, blessing and unwavering support throughout my academic journey. Special thanks also to my fiancée, and I sincerely acknowledge my previous supervisor, who's my now co-supervisor, Dr. Dora Kumar, for his expert guidance, valuable insights and constructive suggestions that significantly contributed to this research and overall of my growth during my PhD tenure. And I express my heartfelt gratitude to my current supervisor, who is Dr. Anupita Ray, for our continuous supervision, scholarly guidelines and encouragement throughout my doctoral research work. And I also thankful to Dr. Nitara Sharma, Dean of our School of Bioengineering and Biosciences, for our academic leadership and institutional support. I also acknowledge our HOD Dr. Ashish Vyas for providing me a conducive research environment and departmental support. And I will sincerely thank Dr. Vishnu Arivarsanal, internal chairperson, and the Professor Prince Sharma, external expert, Punjab University, Chandigarh, for their valuable academic input and constructive evaluation. I also thank my colleagues, Ms. Divya Bajaj, Ms. Ati Turshidwani, Ms. Nisha Yadav, Ms. Anil Basin, for their technical assistance and academic discussion throughout the support. I gratefully acknowledge the School of Bioscience and Bioengineering, lovely professional university, for providing me an excellent research faculty and a support academic environment to grow during my 4 year of PhD tenure. And finally, I thank all those who are directly, indirectly, in any ways contributed to this research, supported me throughout this journey, and I am able to stand today here giving a final viva. And the publications, these are the publications from my PhD work which are published in different journals, and my 2 papers have been published in the Q1 journal which are on impact factor of 4.3, and in the Discover journal which is also Q1 journal, and also in the one journal is the Rasayan Journal of Chemistry, in which also my work has been published, which is a Q3 journal. In both I have published my work in which I have shown the antimicrobial activity and other works. And this is my overall profile, my doodle score profile. So until now the citation is around 381 citation I have maintained in H index of 9, and my total impact factor, cumulative impact factor on my all publication have crossed 70. It's around 71.23 exactly, and I have published 24 publications until now during my PhD tenure, out of which 8 are research papers and remaining are review papers and 2 are the book chapters, and out of them 20 were on Scopus Index publications. And these are the conference certificate which I have done during my PhD tenure, and this one, and this is a oral presentation in Walter conference, and this is the oral presentation in which I presented my research conference or emerging trend and innovation in basic science at Baddi, Himachal Pradesh, and these are the references. Thank you so much everyone for listening me for this much long, and very well thank to you, ma'am.
Thank you, Dr. Now you are supposed to be doctor after the Viva.
Yes.
Now, you have done a very good work.
Yes, ma'am.
You, in this research, you addressed a globally critical health issues like Carbapenem-Resistant pathogens by relating the fungi in nanoparticles induced treatment. But I have few queries.
Yes, ma'am.
So you have to answer. Some questions are from my side and some are from the other expert. So I want to know, why did you select silver nanoparticle over the other metal nanoparticles like gold, zinc for combining carbapenem resistant pathogens?
Yeah.
Why only silver, not the other metals?
Yeah. And during our literature search, when we are finding out topic which nanoparticle to choose, so during our literature search we have find that the silver nanoparticles, and more importantly biogenic silver nanoparticle, has been reported for the broad spectrum antimicrobial activity due to their high surface ratio and this biomolecular capping from plants, fungi and even bacterial. So because of that main reason, and even during our studies we have seen that in gram negative, in even MDR gram negative pathogens, silver nanoparticle has been shown very good antimicrobial activity.
Because it's a well-known fact, we are using the AGNPs.
Yes.
But my main concern is, why can't we use the other metals also? Zinc is also showing antimicrobial activity. In this research, you focused on only one metal, oxide, and you can, when in future you can compare the antimicrobial activity of the other metals against these pathogens also. So second one is, why there are so many pathogens, resistant pathogens, why you choose only E. coli and K. pneumoniae?
Yes, ma'am. The reason behind in the group which we have chosen, the Carbapenem-Resistant Enterobitraceae. In that group, the most prevalent organism and the most prevalent in case of the infection rate, these are E. coli and Klebsiella pneumoniae, and that is the main reason which I have told. So those two organisms are the most prevalent one in that category. It's around 70% the Carbapenem-resistant enterobitraceae infection has been caused due to the E. coli and Klebsiella pneumonia, and they are even show the highest resistance rate compared to other Carbapenem-resistant enterobitraceae, Escherichia, Proteus. That is the reason we have chosen the two isolates.
You brought these pathogens from the PIMS.
Yes, ma'am.
So where you did your research?
Ma'am, I have done at Lovely Professional University Lab.
So what precautions you have taken during this research? Because they are the pathogens, you should be very careful regarding these pathogens. What precautions you have taken for this
Initially,
research?
before doing this work, we have taken the IBSC approval from our biosafety community, and this pathogen has been turned into the risk to group organism. So for that, initially we have used the biosafety cabinets which we have, BSL1 cabinets which we have, and other for the
room is separated from the other room.
Yes, no, it is room is separated. The room is completely
because
separated.
On the fourth floor and it is separately made.
So that's why I want to know, because it is pathogenic,
Ma'am, we
and
have
you have to
different-
take extra care about these pathogens.
Yes, taken all the precautions.
And one, for the contamination part, for the disposal part, we have also followed the proper disposal guidelines which are followed for the disposal of pathogens and the plate which have used during our all the research, all the contaminated part.
So why soil is selected for the source of isolating nanoparticle producing fungi?
Now, basically, as we know
Because
that-
fungi are also available in the other sources also. Why you have taken soil only?
Yes, ma'am. Ma'am, actually, soil has been the soil, and basically we have taken an agriculture soil temperate because it is more rich in organic matter. Na? When more able to get the more better, more highly growther fungi compared to the normal soil. And ma'am, we have taken endophytic fungi. Ma'am, more research has been done in the endophytic area. There is more research has been done. Soil is, ma'am, basically under explore in case of getting the MDR pathogen. This is the mainly.
There are papers about the fungi from other sources also.
Yes, ma'am. There are majorly, ma'am, there are endophytic fungi.
You found any difference in the fungi from the soil or the other sources nanoparticles?
There is not much difference.
Okay. So next is what advantages does extracellular synthesis over intracellular synthesis of the nanoparticles?
I mean, both the synthesis we have seen that majorly the reported literature have shown the silver nanoparticle synthesis from fungi are mainly intracellular. No one reported extracellular synthesis, and in our cases we are initially finding that we are not able to get the extracellular, but we have tried it, and from out of the 25 isolated, only 2 have been able to produce extracellular synthesis. When both are enriched in the extracellular metabolites. And
so what's the reason कि extracellular जो है activity it is less as compared to the intracellular?
Basically the main reason is the component which are involved in the reduction and biostabilization of this nanoparticle are more found in the intracellular extract compared to the extracellular extract. This is the main reason, because of those molecules we are able to synthesize the silver nanoparticle, and the stabilization is mainly depend on that.
So what is the importance of zeta potential in determining the nanoparticle stability?
And zeta, in the zeta potential, based on the isoelectric point, we have checked that the stability, how much it is stability in the liquid medium. So that's what we have checked. I mean, mostly, ma'am, the silver nanoparticle which are found in the literature are showing -28 to 30, and because they are highly stable and moderate. But in our cases, we found it are moderately stable. But still, it is okay because it is small in size and moderate.
Otherwise, zeta potential is used for the stability of the nanoparticles.
Yes,
but in your case, they are moderately stable.
Moderately stable, ma'am.
And what are the functional group identified by the FTR IR in nanoparticle stabilization?
And there are certain groups like certain peak which you have seen. I can show you. In this we can see that certain amide groups are also involved, because we need the certain amide groups are also involved, certain CH groups are also involved.
So these are the functional groups.
Yeah, these are the functional which are involved either in the stabilization, reduction of the silver nitrate for the formation, overall biosynthesis. These are the functional groups.
What evidence supports the multi-target mechanism of action of AGNPs in your study?
And basically in our study we have found that our silver nanoparticle has been inducing multi-target mechanism. They are inhibiting they are damaging the cell wall, they are damaging the cell wall, cell membrane, they even damage the DNA inside the cell, they are even reducing the overall lag phase which is inhibiting the growth. And because of that, what happened when in the case of resistance, the antibiotic which you make has a certain mechanism, either they inhibit the DNA, either they inhibit the cell wall. But if in our case of nanoparticle is making the multi-target, in which we are able to target it more group of the resistant mechanism.
What is more damaged by the bacterial activity, whether it is cell wall or DNA?
Cell wall basically.
Cell wall, because DNA is present in the nucleus, it is least affected with the activity. So how was the fungal isolate identified as Aspergillus niger at the molecular level, and why are ITS primers commonly used, not other primers? Why you use ITS primers?
Because these primers are consistent in the organism. They are consistent sequences which are present in the organism. That's why we use ITS1 and ITS4 primers. So
other primers can also be used or not?
They can also use them.
Can be used. Okay. So, can prolonged exposure of silver nanoparticle induce resistance in bacteria?
Basically, there are certain papers which have coming out now in which.
But you didn't test this.
I have not tested, but there are heavy metal because there are certain effluxum present in the bacteria which treated this heavy metal as a toxic compound, and they throw down. Because of that, there are certain metals, not silver basically.
But in your study, you didn't include this. Okay, how would you design an in vivo infection model to validate the antibacterial efficacy of your agent?
And for that, we have to design certain animals models, certain models in which we.
Which animal model you can use? use?
Basically the reported are basically mouse models and
In literature, which animals used by the other researchers?
such as rat and mouse models basically.
Okay. Not Drosophila insect cannot be used?
They are not using, they are not published until now.
Okay. What are the possible environmental implications of large-scale AGNP's production and applications?
And basically at environmental scale, they are, as we seen that we are come with the biogenic silver nanoparticle synthesis as it is less toxic compared to the physical and chemical method we have used. The environmental implication in as not much we can see that they are not damaging environmental in even large scale. If you are doing that, I don't think so. There are heavy metal toxicity which can happen at environmental scale at the soil, that can happen.
What are your recommendations regarding the fungal mediated nanoparticle synthesis for the industrial production?
And for the industrial, as we can say that in case of we are not using bacteria and we are using fungi, and not plant even, because in case of fungi we can say they are consistent, they are more reproducible, and they are presently high biomass. Because of that, in the industrial scale, at high scale, we can also use fungi for standardize this process to yield more and more nanoparticles.
You filed any patent for this?
No, ma'am, we are not finding any patent.
There are patent regarding this available or not?
And that I don't know.
Because madam, you can file the patent if you have designed the fungal mediated AGNPs. If there is no patent, you can apply for this, I think. Because otherwise we can use this research for the health purpose. So we can industrialize this research to the industries also. Transfer the technology to the industry also.
Basically, right now we are in the process of making an ointment out of our silver, using our silver net.
But you can found and try for this.
Yes, ma'am, definitely I will find one.
Okay, this is all from my side, and there are some questions from the other expert. So you made all the changes in your synopsis?
Yes, ma'am, all the changes.
Because he or she recommended certain revision in your thesis.
Yes.
You did?
Yes, ma'am, I have done all the changes.
So there are few questions. What is the advantage of fungal mediated synthesis of silver nano over the chemical synthesis?
Yes,
the same I think I have asked. Why did only limited number of fungal isolates successfully synthesize the nanoparticles?
And this is the reason of because there are certain reducing compounds which are not present in the each and every fungi. Like if I talk about certain enzyme like NADPH reductase which are involved, that are not present in all the fungi. Certain biomolecules which are reduced and they are not present in all the fungi. That is the main reason. Yes, only the certain amount of fungi are produced.
Maybe laboratory conditions can also be responsible for this.
Yes, ma'am. Sorry.
Some laboratory environment or conditions can also.
No, ma'am. We have tested them under the control condition, control pH and temperature.
So one thing more, why you recharacterize the pathogens?
And basically, when we transported them from the lab, so there are the chances of the contamination. In microbiology, there is the one thing we have taught always is contamination. So we have to recharacterize them. So no issues will be when we have performed our experiment. There will be no error.
So how does the MIC-MBC ratio indicate bacteriostatic and bactericidal activity? This is the question.
Yes, ma'am. So when as we compare our MIC and MBC ratio, we have compared the ratio which we are getting is compare our just. No, ma'am, here the ratio which we are getting from our, in case of E. coli, the MBC or MBC ratio which we are getting is comparatively high, which are shown as a large window for the therapeutic dose, ma'am.
Okay. What is zeta potential I have asked? How do silver nanoparticle induce DNA damage in bacteria?
Sorry, ma'am?
How do silver nanoparticle induce DNA damage in bacteria?
And basically, they have disrupt the membrane and they have enter the nucleus and then.
So there are certain barriers when they enter into the nucleus. Yes, so I think it is very less silver nanoparticles can enter into the nucleus because there is a nuclear wall around the nucleus. So they can destroy the cell wall as well as the cytoplasm, but in the nucleus the DNA damage is very less and they can't completely damage the DNA.
That we have also shown in our study. And there are two things. One thing is they completely rupture the cell wall. Another thing there is due to
otherwise nucleus also have the nuclear DNA damage repair system also. So I think this is very less tendency to damage the DNA of the bacteria. Yes, so what are the limitations of your study addressed in future research?
Yes, ma'am.
Which you can improve?
Yes, ma'am. These are the limitations in vivo studies and all.
How can this nanoparticle system be translated into clinical applications? I have asked, I think,
Yes.
how you can apply. So these are the questions from my side as well as from the other experts. This is all from my side, and congratulate you from my side, and
Thank
you
you,
can
ma'am.
also ask.
Okay, Himanshu,
Yes.
can you just tell me what kind of work you are planning for after this PhD submission?
Sir, after this PhD submission in the postdoc, I am trying to thought of do the molecular identification of this gene, because if we do it at the genetic level and we are identify which gene is causing the resistant infection more, then we are able to make the result, the clinical application will be more better. So we are if I identify the particular genes in case, and the main case is in vivo studies which I have to do, sir.
Okay, so as ma'am has told that you have not filed for any patent.
Yes, sir.
So do you have any idea that how can you put patent on this silver nanoparticle?
Yes, sir. Initially, sir, actually due to the time constraint, we are not able to do it at the last time. We are designing a hydrogel, and we are incorporating these silver nanoparticle into that and use them for the wound infection healing. Because as we see that,
But
yes,
that will take an extra PhD. You can't do one more PhD.
From this work,
With this work, what's the plan? How will you do patent for this?
so for this
From your
work,
thesis.
so we can patent our processor, basically the process which is used for synthesizing.
That you will do. I don't mind.
That right.
But if you do hydrogel work, that will become another PhD.
Yes, sir, definitely, sir.
There's no time for doing other.
Yes, sir, that
you want to pursue this research in future?
Yes, ma'am, I want to.
In which form?
Ma'am, in
Postdoctorate?
Yes, ma'am.
In same university or any other university?
Ma'am, so ma'am, I have to see, because there are certain facilities which I have required. If I do the higher level of work, then certain facilities I have required, and because more is the genetic level of work I want to do in this part, and for that there are certain advanced laboratories. There is a certain facility which is not here, which we can come up with. If there is a project, we can establish that facility. So that's the cost constraint and resource constraint there, ma'am.
Okay, that's all from my side. Dr. Alok.
Yeah, and also good work actually. Just a simple question: so why we have selected P85 cell line in order to fit this cycle?
So the 3D series cell line is the most standardized cell line, and most in case of when we found the literature, the test in silver nanoparticle, they are using 3T3 cell line. So they are the most standardized cell line for nanotoxicology. So we have used the cell line. So that's the main reason.
Any other cell line can be used?
Many other cell lines can be used, sir. But these are the most standardized, and because sir, we have to do only the preliminary toxicity evaluation of a nanoparticle. So we do with this standardized cell line. If we have to do the more toxicology study, we can use different cell line, different protocol, different concentration which I use, and we can do the more deeper toxicity evaluation we can do with this.
Other question we have already discussed in the ATP.
Okay. Ma'am, if it is okay, then we can proceed with your result.
Yeah.
Whether we can call him Dr. Himanshu.
No, we can call him Dr. Himanshu. Congratulations and best of luck to your future.